We're going to transition now from general physiological mechanisms to specifics about neurotransmitters. So this table has a bunch of specifics. And I don't like talking about text. I like talking about pictures. And this is just text. So I'm going to leave this, so you have this text as a handout, as a resource to refer to. So use this table and the next table as a reference table. And I'm going to move on to the next PowerPoint real quick. So the neurotransmitters and receptors we're going to talk about for the next hour. Acetylcholine, which you've already been introduced to. Dopamine, which you haven't been, but are going to. Norepinephrine, which some of you have heard of a lot of from the other A&P course. Serotonin. Some of you have learned about serotonin in other courses. Glutamate, an excitatory neurotransmitter. And GABA, which you've learned about from the problem set. We're going to skip glycine, so you can just kind of cross that one off of your table. We're not going to get into it. And we're not going to talk about purines, so you can cross this one off your table. But the other ones we're going to talk about now in detail. And then you can refer to this table as you study the neurotransmitters. So I'm going to open up the other PowerPoint. So we're transitioning to the second PowerPoint. And we're going to talk about neurotransmitters, and we're going to talk about some neuroanatomy. So here are the tables again. So let's start talking about acetylcholine. I will not ask you to draw the structure of acetylcholine, but there it is. Acetate, for those of you who are chemists, acetate choline, acetylcholine. It is the one neurotransmitter that we're going to talk about that is not taken up intact. It's hydrolyzed in the synaptic cleft. So I'm not going to test you on this factoid, but the factoid is that acetylcholine is hydrolyzed by esterases, acetylcholine esterases that are in the synaptic cleft of cholinergic synapses. So in cholinergic synapses, there are cholinesterases that hydrolyze acetylcholine. So it is not. So its removal is a combination of hydrolysis and uptake. All the other neurotransmitters we're going to talk about follow the simple mechanism that we talked about a few minutes ago of reuptake and repackaging intact. So here's a cartoon of the nicotinic acetylcholine receptor. Synaptic cleft, extracellular, transmembrane, intracellular domains. Now looking down the pore, so looking from above down the pore, you can see it's a pore. It's a hole through the membrane, but there are binding sites that are specific for sodium and potassium that allow it to be a cation channel. So nicotinic receptors are cation channels, and they cause depolarization because of the sodium influx. Acetylcholine also binds to a different class of receptors called G-protein, GPCRs. So there are acetylcholine GPCRs, and they're called muscarinic receptors. So the nicotinic receptors are ion channels, and the muscarinic receptors are GPCRs. I do want you to know that language. Where do these names come from? It comes from pharmacology. Nicotine, the drug you've heard of, activates these receptors. Muscarine is a toxin in mushrooms When people die of mushroom poisoning, they die because they eat this toxin, and it activates these receptors, muscarinic. There are multiple types. I do not want you to know the names and the G proteins that are coupled to the different types of muscarinic receptors. If I want you to tell me a story, I will tell you, start with GQ. I do want you to know the GQ story, but I do not want you to memorize that M1, M3, and M5 subtype of muscarinic receptors are coupled to GQ. I just told you that, but don't devote brain space to it. Know the GQ story, know the GI story, but don't memorize which ones are which. Is that clear? Now we're going to transition to another class of neurotransmitters, the monoamines. And there's going to be two types, so I'm introducing the language of these neurotransmitters. There are two types of monoamines. There are the catecholamines, and there is serotonin. The catecholamines are dopamine, norepinephrine, and epinephrine. What makes them a catecholamine is this thing right here. Benzene ring with two hydroxyls in the ortho position. That makes this a catechol. So a little bit of organic chemistry. Did you get this in organic? Maybe. You certainly got ortho, right? So ortho-hydroxy benzene ring makes this a catecholamine. Monoamine. So dopamine, norepinephrine, and epinephrine are the catecholamines. I do want you to know that. And serotonin is a monoamine, but it is not a catecholamine. You'll see it in a minute. So this is dopamine. Dopamine is a catecholamine. It's important for many things, including motor. We'll talk when we get to the motor system. You're going to see dopamine and dopamine receptors. Our cadaver died of Parkinson's disease, which is caused by a destruction of the dopaminergic neurons in a part of his brain. So depletions of dopamine cause Parkinson's. And the gentleman who gave his life to our study died of that. So it's important for motor control. It's important for the etiology of Parkinson's disease. It's important for addiction, which is a pathological excess of the reward pathway. We're going to talk about that in a minute. There are the home of many, not all, but many, of the dopaminergic neurons is in an area of the brain, two areas of the brain, called the substantia nigra and the ventral tegmental area. That's neuroanatomy. I'm introducing some neuroanatomy that I do want you to know. Dopaminergic cells that release dopamine have their cell bodies in these areas of the midbrain. Just, I'm using the language. The terminals of those, so that's not where the dopamine is released. That's where the cell bodies of those neurons are. They send axons pretty much everywhere. And you can see why, if you destroy this area of the brain through Parkinson's, and it's sending dopamine everywhere, why someone might develop dementia, Parkinsonian dementia. Some of these areas are motor control areas, so Parkinsonian abnormalities in movement, and death. because it's so everywhere. Dopamine, but here's the key point, dopamine is released everywhere, but the key parts of the cell bodies where the neurons that release it live, where their cell bodies are, is the substantia nigra and the ventral tegmental area in this part of the brain called the midbrain. Using some neuroanatomy. Just to get oriented, I hope you already can see that this is a sagittal section of the brain. We've cut the brain in half, and we're looking at half of it. So there's half of the cerebellum, there's half of the cortex, there's half of the brain stem, there's the midbrain, substantia nigra, a part of the midbrain. Dopamine is a derivative of tyrosine. In fact, all of the catecholamines, dopamine, norepinephrine, and epinephrine, are all derivatives of the amino acid tyrosine. Because there's the ring, and there's the catechol. Catechol. I do not want you to memorize the names of the enzymes that do this synthesis. I do not want you to memorize the order. I just want you to know that all of these catecholamines are derivatives of tyrosine. Dopamine receptors, like the previous slide. Dopamine receptors, the receptors are everywhere because dopamine is released globally in the brain. There are two types of dopamine receptors. They are both GPCRs. I don't want you to memorize what D1 and D2 are. I don't want you to make the relationship that this is a GS coupled one and this is a GI coupled one. Don't do that. But do know that they are all GPCRs. There is no such thing as an ion channel receptor for dopamine. We don't have it. We didn't evolve it. So it's important so going back to the table, you look at your table and you say dopamine, okay, it says motor control. There is a section, there is information going from the substantia nigra to the striatum that is important for motor control. We'll see that in more detail when we get to the motor system. I'm not going to test you on that now, but I'm going to reinforce that when we get to the motor system. Dopamine is important for reward and addiction. There is neurotransmission from the ventral tegmental area to an area called the nucleus accumbens. So that's what's cartooned here, dendrites in the nucleus accumbens. So here I'm not going to ask you to identify the nucleus accumbens because it's tiny and it's hard to see. But there is dopaminergic neurotransmission from the ventral tegmental area to the nucleus accumbens that is important for reward. Whether the reward is mating or eating or winning a game or drinking if you're thirsty. Those are all rewarding events. Drinking if you're thirsty, eating if you're hungry, mating if you're a mammal. Those are all rewarding events, and they change the distribution of dopamine receptors in the nucleus accumbens. This reward pathway goes crazy during addiction. It goes to excess. So cocaine, heroin, other addictive drugs activate the reward pathway And people get addicted because they say, whoa, that was nice. I like that reward. I like that feeling. That feeling is good. I want more of that. And that becomes addiction in susceptible individuals. And the susceptibility to addiction is an interesting variable. Some of us could try cocaine and not get addicted. Some of us could try cocaine once, and it's life-changing because it would trigger addiction. Some of us used to be addicted to nicotine. Nicotine is another addictive drug that activates dopaminergic pathways. So drug addiction and reward pathways are related, one being physiological and the other being non-physiological. So the other thing that dopamine receptors are important to, Parkinson's, depletions, and schizophrenia. I'm not going to ask you too much about disease on any exam, but I might ask you to know motor, reward, disease. Question? The neuron, so the question is addiction associated with an irreversible change in dopamine receptors? And the answer is yeah. It can. So activation of those receptors can cause changes in synapses, changes in expression of neurotransmitter receptors, changes in behavior. So the question is, someone who was addicted and is able to recover from addiction, is their brain still changed, or is their brain simply overriding so are they still compulsively attracted to, let's say, cocaine or heroin or alcohol or nicotine? But typically there could be reversions in the reward pathway back to normal, but maybe not all the way. And there are changes in other behavioral pathways that are brought into play and become more powerful than the draw of the reward pathway. So people learn, oh, I smoke that cigarette, I'm going to get screamed at by my spouse. That's not fun. That's a negative stimulus. Let's not do that. The cost starts to have its impact on your neurobiology. Negative, the inverse of reward. Hannah?
But the urge is still there.
Well, I can only speak for myself. yeah, I think so. I mean, if I watch someone smoke a cigarette, I want a cigarette. Still, in years. Okay. One more question. Okay. So now let's transition from dopamine to norepinephrine, another catecholamine. Like dopamine, there are cell bodies in the brainstem in an area called the locus ceruleus. So now your question is, do I need to know the name substantia nigra and ventral tegmental area? Do I need to know that those are associated with dopamine? Yes. Do I need to know that the locus ceruleus, which is located very close to those other areas, are where norepinephrine neurons have their cell bodies? Yes. And just like dopamine, the axons from the locus ceruleus go everywhere. And the norepinephrine in the central nervous system is important for things like arousal, waking up, being just physically aroused, not asleep, not unconscious. And they are going to be important in the sympathetic nervous system, which we're going to talk about, I guess, on Wednesday. There are multiple types of epinephrine and norepinephrine receptors. So epinephrine and norepinephrine share receptors. They are all G protein coupled. There are no ionotropic, ion channel linked receptors. They're all G protein coupled. They are all 7 transmembrane helices. That's what this cartoon is illustrating. They're coupled to different G proteins. They do important things. I do not memorize, this is for reference, do not memorize what the alpha-1 receptors do, what the alpha-2 receptors do, what the beta-1, beta-2, and beta-3 receptors do. Do not do that. But do know that different GPCRs are coupled to different G proteins, and know the stories of those G proteins and the things downstream from them. So if I ask you a question about beta receptors, I will tell you in the question that beta receptors are coupled to GS, and then ask you to work on a problem related to that knowledge. I do not want you to make this knowledge. I do want you to know what happens downstream from GS and be able to apply that knowledge. Okay? So I'm trying to, there's just a ton of information, and there are things that I want you to know, and the things that I don't want you to know because it takes up more brain space than it's worth. Zoe?
What is a deficiency and an increase in norepinephrine associated with, like we talked about how dopamine is associated with like Parkinson's, when it was in depletions, there are like correlations to diseases with
So depletions of norepinephrine. So it's complicated, and it's not as straightforward as dopamine. So and I don't want to bullshit it. I don't want to make things up. But abnormalities in sleep pattern are sometimes downstream from abnormalities in norepinephrine signaling. Abnormalities in temperature homeostasis are sometimes downstream from abnormalities in norepinephrine homeostasis. Some mood disorders are downstream from norepinephrine homeostasis. So some of the antidepressants that we learned about affect norepinephrine signaling. We're going to get to that concept with serotonin real soon. But some of them are related to norepinephrine. But there's no disease. There's no disease for norepinephrine the way Parkinson's is for dopamine. Michael?
So we talked about this in AMP2 a lot. Does like the sleep arousal, pain, and all that, is that all from like stroke volume and the heart rate? Because how like norepinephrine can control your heart rate and all that stuff.
So norepinephrine acting through beta-1 receptors is what controls your heart rate.
Is that what causes you to like how we're talking about like the sleep and stuff and your arousal? It's like
So alpha and beta could be associated with arousal.
okay.
And alpha 2 could be coupled with, the reverse of arousal, the settling down.
Okay.
Question?
Yes, so I do think this is what you're referring to,
Both are the adrenergic receptors for epinephrine or norepinephrine, and the answer is both. And some types of these are more specific for norepinephrine, and some types of these are more specific for epinephrine. But that's a neuropharmacological detail that we don't need to get into. So epinephrine and norepinephrine are the adrenergic neurotransmitters. The jargon here is weird because In England, adrenaline and noradrenaline, for some reason in the United States of America, we call those two neurotransmitters, we don't call adrenaline a neurotransmitter. Adrenaline is a drug in the US, but it's epinephrine. Epinephrine is adrenaline, norepinephrine is noradrenaline, so we talk about adrenergic neurotransmission by epinephrine and norepinephrine in the US, by adrenaline and noradrenaline in the UK. Sorry, it's just language, so it has some dialect. Did that answer your question, Berto?
Yeah.
Okay. Serotonin is a monoamine, but it is not a catecholamine. It is a derivative of tryptophan, not tyrosine. I do want you to know that. The location of key serotonergic neurons, their cell bodies are in an area of the brain, midbrain, called the Raffi nucleus. I do want you to know that. So, substantia nigra, ventral tegmental area, dopamine. Locus ceruleus, epinephrine and norepinephrine. Raffi nuclei, serotonin. Serotonin is important. So, again, GPCRs, and there is one form of a serotonin receptor that is a ligand-gated ion channel that is in fact very similar to the nicotinic receptor. So two types of receptors, ionotropic and metabotropic, GPCR. You know that serotonin is important for mood and other things because the SSRI class of antidepressants act by inhibiting the sodium, this is wrong, the sodium neurotransmitter reuptake mechanism. The target for SSRIs is the serotonin reuptake transporter. So by inhibiting this transporter, there is more serotonin in serotonergic synapses, and that becomes antidepressant. The mechanisms are complicated, actually. But you know that serotonin is important for mood by knowing that selective serotonin reuptake inhibitors improve mood for someone who is depressed. Let's talk about, I just want to mention that all of the neurotransmitters we've been talking about, acetylcholine, the catecholamines, serotonin. We haven't talked about glutamate and GABA, but they also are small molecules. There are also peptide neurotransmitters. And maybe let's just leave it at that. There are peptide neurotransmitters. So now referring to your table, glutamate and GABA are neurotransmitters that are released everywhere. Their cell bodies are everywhere. There's no picture I can show that makes it look pretty. I mean, that is interesting to look at. But I do want you to know that glutamate, the amino acid, is a neurotransmitter that is the most important excitatory neurotransmitter in our nervous system. And I do want you to know that GABA is the most important inhibitory neurotransmitter in our nervous system. And your problem set already asked you to work with GABA receptors. GABA A receptors are chloride channels that you worked on your problem set, so that's knowledge that you have already integrated and are using. And I do want you to know that GABA B receptors are G protein coupled receptors that are coupled to GI and cause It's like that autoreceptor story I told you. They're coupled to GI, increasing K channels, hyperpolarization. So there's two ways we can hyperpolarize a neuron with GABA. GABA A receptors and chloride influx, GABA B receptors and potassium efflux. Both of those hyperpolarize, that's why GABA is inhibitory most of the time. That's a whirlwind view of the neurotransmitters and what I want you to know and what I don't want you to know. Your job now is to use, to read, to take your notes and to write your notes and to integrate the things that you need to know. That's your job. I know it's hard work because it's a lot of information, but integrating that information for the exam on Monday, a week from now. Well, less than a week from now, but a week from today. What I want to do now is spend the last, half hour, maybe 40 minutes, introducing some language of neuroanatomy. We're going to build on this on Wednesday. So I want to introduce it and then reinforce it on Wednesday. This is anatomy. It is not on the quiz. It is on the exam. Okay, so we're looking forward now. Even though it's anatomy, it's not on the quiz. So here's a cartoon of a sagittal section. Midline, sagittal section, looking at the brain from the middle. Let's talk about some of the structures, just using the names. Spinal cord is connected to the brain by the brain stem, which has three regions. The medulla oblongata, the pons, and the midbrain. I do want you to know these words and their order, which one is which. The pons is Latin for bridge. Some of you may know pontine, as an adjective that describes a bridge. It's a bridge between the brain and the cerebrum and the cerebellum. So the cerebellum is connected to the brain, to the cerebrum, via the pons. That's the bridge. The cerebrum has multiple names, and obviously I'm not going to ask you to know every single neuroanatomical term. There's thousands of them, but I want you to know some of the most important ones. The frontal lobe, the parietal lobe of the cerebral cortex, the occipital lobe of the cerebral cortex, the temporal lobe of the cerebral cortex. That's neuroanatomy that I do want you to learn. If we cut open the brain and look at the midline sagittal section, there's an area here called the thalamus. So I want you to annotate your slide and write the word thalamus. Thalamus. A structure called the corpus callosum, which is white matter that connects the two hemispheres. So this is the right hemisphere of this person. This is his left hemisphere. The corpus callosum connects those two hemispheres. The next slide has some more detail that we're going to work on. And the cerebellum. So you do want to learn some neuroanatomy. That's why you're taking this course. And I'm going to try to make them functionally relevant too, by telling you what each part of these brains, what each parts of the brain does, at least in part. So here are some images of a real brain. So in the past, last year, pre-COVID, I handed out a laser pointer, and the laser pointer was handed from person to person, and the person pointed to something and called out a name. right? It was your choice of what to point to, and because that's what you knew the name of. Well, we can't do that. That's against the rules. So I'm going to point to stuff. And I'm of two minds whether I'm going to call on people around the room or whether I'm going to ask for volunteers. So, I'm going to give you the option. I'm going to start with pointing to something. Well, no, actually, since you haven't learned it yet, I'm just going to talk the language. And then on Wednesday, I might come back and do the game where I'm going to point to something and say, Ramsey, what's this? And then Hannah, what's this? I might actually put you on the spot. So be prepared for that on Wednesday. So do the quiz today, then Tuesday prepare for this sort of game. It won't count for anything, but it will help reinforce knowledge. So let me just talk it through for the first time. Lateral view from the side. Sagittal section, sagittal view. Superior view, looking down on the brain. So this is the front, and this is the back. This is the anterior, posterior, top. Okay, let's look at this piece right here first. This, did I give this picture to you as a handout? No. Okay, so you just have to write somewhere in the margins. This is the longitudinal, yeah, go ahead and cut those lights. Yeah, that's better. This is called the longitudinal fissure. Longitudinal fissure. Right hemisphere, left hemisphere. Frontal lobe, parietal lobe, occipital lobe. The generic name for this lump of tissue, remember the brain is all folded, so we've got outfolds and infolds, right? These outfolds are called, one of them is called a gyrus. So the gyruses are the lump, and the folds between the gyruses, which are really called gyri in Latin, two gyri are separated by a sulcus. So that's an anatomical name. Gyrus and sulcus. So this is a gyrus, and this is a sulcus. Gyrus, sulcus. Gyrus, sulcus. Some of these gyruses and gyri and sulci are going to have names that I will want you to know, and some of them won't. Okay? Lateral view. Frontal lobe, parietal lobe, occipital lobe. Occipital, parietal, frontal. Temporal. This is the temporal lobe. This here, that separates the temporal lobe from the frontal and the parietal lobes, this sulcus right here, is called the lateral sulcus. This sulcus that separates the frontal lobe from the parietal lobe, this sulcus has a name that I do want you to know. It's called the central sulcus. Frontal lobe, central sulcus, parietal lobe. The gyrus in front of the central sulcus is called the precentral gyrus. Name is kind of helpful. Before. Precentral gyrus. It's part of the frontal lobe. because it's pre to the central sulcus. This gyrus here, I bet you know what the name of that one is already. It's the post-central gyrus. And it's part of the parietal lobe because it's post of the central sulcus. Frontal lobe, pre-central gyrus, motor. It's going to have motor function. Sulcus, central sulcus, post-central gyrus, sensation, somatosensation, sensory cortex here. We're going to see that when we get to this next section of the course. Occipital lobe, parietal, occipital. We'll talk some more about some of the structures here later. Cerebellum. Okay, let's look at this midline sagittal section. Medulla. Pons. The connection to the cerebellum. Midbrain, where the ventral tegmental area, substantia nigra, locus ceruleus, and raphe nuclei are. Midbrain. Brain stem together. Thalamus. Corpus callosum. What lobe? Frontal lobe. What lobe? Parietal lobe. What lobe? Occipital lobe. What lobe? Temporal lobe. We're seeing the sort of the inside of the temporal lobe. This right here, so under the corpus callosum is an area, is a membrane. Behind the membrane and in front of the membrane are two ventricles. The ventricles of the brain, not like heart ventricles, well, like heart ventricles, it's a space. The ventricles of the brain are where cerebrospinal fluid are. And this is behind this membrane is the lateral ventricle. So we've got two lateral ventricles behind the membrane and on this side of the membrane. We're seeing the membrane that separates the two lateral ventricles. There's going to be some other ventricles that we'll talk about as well. Ventricles are, they're filled with fluid, not with neurons. But it's important. Does anyone know the name of this gyrus right here? It's got a name. Some of you sounded like you knew some neuroanatomy already. Anybody know the name of that one? It's called the cingulate gyrus. Not singular, but cingulate. Cingulate gyrus. It's an important gyrus of the limbic system. It's important for emotion, fear, anger, repulsion, part of the limbic system. Over here, what lobe? Occipital lobe. There's a gyrus here. It's got a name that's very helpful. The parieto-occipital sulcus. The sulcus that separates the parietal from the occipital. That one's easy. There's a gyrus here that has a different name. It's called the calcarine sulcus. Calcarine sulcus. And why am I telling you that one? Because the gyri on either side of that sulcus is the visual cortex. It's going to be important for vision. So you can see I'm using some of the functional names. So the next slide, the one that I did hand out, gives you the list of things that I want you to, so this is your decoder key. I know it's too small to read on the handout, but on the slide if you go to Moodle, you can read these things. So, A, some central sulcus. So this is written in Latin, but you can translate the Latin to English. Sulcus centralis is the central sulcus. I do not, this is what you don't need to know, but you can play the game and learn more than I want you to know. I mean, that's fine. But, so A is the central sulcus. Where's A? A, central sulcus. Central sulcus. Oh, that's weird. Where's A? So, I'm expecting it. What is Q? Okay, this is weird. I shouldn't have. B is, so I don't know where A went. The A tag must have fallen off when they took the picture. Because B is labeling the lateral sulcus, the sulcus lateralis. Anyway, you can play the game and learn those, but what I do want you to know is here. These are terms I want you to know and to be able to identify on an image. Point to a sulcus, any sulcus, and point to the central sulcus. Point to a gyrus, any gyrus, or maybe point specifically to the post or pre-central gyrus. Know the longitudinal fissure, which is not shown here because it's been cut. Well, the longitudinal fissure is where this was cut. The lateral fissure or the lateral sulcus, same thing. Lobes, frontal, parietal, occipital, temporal, cerebellum, midbrain, the brainstem, the thalamus, and the hypothalamus. Hypo, below, thalamus, hypothalamus. Corpus callosum, cingulate gyrus, and we can't even see, we're going to talk about the hippocampus, but we can't see it on this image. It's not on the surface, and it's not at the midline. It's lateral. We'll see it in a minute. So those are things to start to, that's anatomy language that I do want you to know. We're going to reinforce that in lab, because we're going to be looking at the brain and using these words in lab to reinforce the knowledge. But, study. So this is a sagittal section. We could make a coronal section. And so here is a cartoon of the brain in coronal section. What's the name of this sulcus right here? Longitudinal sulcus, longitudinal fissure. What's the name of this lobe right here? It's on the sides, it's lateral. So you're going to have to start to see three-dimensional objects in two-dimensional sections. The temporal lobe from the side, the temporal lobe cut in a coronal section. So what's the name of this sulcus right here and this sulcus right here? The lateral sulcus, the sulcus that separates the temporal lobe from either the parietal or the frontal lobe. Thalamus in the middle, corpus callosum, hypothalamus below the thalamus. We're going to talk about the pituitary a lot when we get to the endocrine section, but just introducing the word here. The one thing from this slide that I do want you to see, but I won't test you on, I will not test you on, is the layered structure of the gray matter. So we're going to see gray matter, which is where cell bodies and dendrites are. And we're going to see white matter where axons are, where myelinated and unmyelinated axons are. So white matter is myelinated and unmyelinated axons, the connections, and gray matter is where the cell bodies and dendrites are. The gray matter of the brain has a layered structure. It's a highly organized, layered structure. I am not going to ask you to know the different layers and what cell bodies are in which layers. But I do want you to know that there are layers. And there are layers defined by what cells, what cell bodies are in what layer. But I'm not going to ask you to memorize that. So let's look at a coronal section. I gave you this as a handout. So let's spend the last 10 minutes or so labeling it. We can do that. I think I want to see how much you already know and haven't yet forgotten from the language that we used. So let's go around the room. Ramsey, what's the name of this lobe? Temporal lobe. So label your handout temporal lobe. Hannah, what's the name of this sulcus right here and here? The lateral sulcus. Eric, what type of matter is this? White matter. Connor, what type of matter is this?
Gray matter.
Gray matter. Yeah, some of these are easy, right? Don't assume that I'm asking you a trick question if it's not tricky. Okay, Sarah, what is the generic name for this? This here is a sulcus. This is a groove. This is a gyrus, and this is a sulcus. So gyrus, sulcus, gyrus, gyrus, sulcus, gyrus, sulcus, gyrus. Okay? Okay, Elena, what's the name of this sulcus? Longitudinal fissure or longitudinal sulcus. Okay, Henry, this is a challenge question. I want to make everybody know that this is a challenge question. What's the name of this gyrus? Okay, I'm going to give you a hint. I'm going to give you some help. So remember, there's this structure here that you should be able to name, and the gyrus right above it has a name. Right? The name is on the board. What's the name of this gyrus right above this thing right here?
Is it the cingulate gyrus?
The cingulate gyrus. This is the cingulate gyrus. Okay, Andrew, what's the name of this structure right here? The corpus callosum. Okay, Olivia, what's the name of this space that is filled with fluid? Nope, it's a space. So it's the space behind this membrane, right? If we cut this membrane like this, there's the membrane, there's the space behind it or in front of it. Lindsay?
Lateral ventricles.
Lateral ventricles. These are the lateral ventricles. And I'm going to tell you the name of the membrane that separates the two lateral ventricles is called the septum pellucidum. I'm not 100% sure. One L? No, like this. Two L's. Septum pellucidum. Okay, let's see. What else can we see on here? Now, okay, who's next? Andrew, okay. We're going to cycle. Oh, we're coming back. Olivia, Lindsay, Michael, right. what's the, so I'm gonna ask, so don't write down the word he's gonna say, because he's gonna tell me the wrong answer, but tell me what this is.
I mean, it's like the thalamus. but-
Yeah, that's the wrong answer. So this, remember, that, so this cartoon makes it seem like this thing in the middle is going to be the thalamus. But the thalamus is back here. And this section we're looking at is up here. So I have to tell you if that's tricky. So I would have to tell you, if I asked you to label this section, I would say this is a section anterior to the thalamus. Because then you would know that this is not the thalamus, and this is the basal nuclei. We're going to talk more about that. We're going to unpack that in great detail on Wednesday. So let's see, are there any other structures that I could ask you to name on this coronal section? Okay, knowing that this is not the thalamus, that this is the section in front of the thalamus, not at the thalamus, but anterior to the thalamus. Because if the section were here, then Michael would be right that this is the thalamus. But the section is not here, the section is here, and so it's not the thalamus. Knowing that, who's next? Jenna. Knowing that, what's the name of this lobe?
Frontal lobe?
Frontal lobe. If this was the thalamus, okay, next person, Sam. If this was the thalamus, if this was a section that was through here, what would be the name of this lobe? Parietal. Right? So if I have to tell you where the section is in order for you to label it correctly. This is a section through the frontal lobe, not through the thalamus or parietal lobe. Let's stop here. That's good for today. Lots of information that you need to pack into your brains. Remember, the quiz is due at 9 p.m. today. Some of you may have already taken it, I don't know. But it is due at 9 p.m. today. It will not include neuroanatomy, but it will include the anatomy coloring book assignments and the worksheet that we worked on last week. That's what's on the quiz.